Let's talk about transgenic production in plants. Transgene, what is it? It is a transfer of a gene to an organism via recombinant DNA technology. What is recombinant DNA technology then? It is construction of a recombinant DNA, and often this DNA is cloned. So recombinant DNA, it is the foreign DNA that is inserted into a vector. So what's a vector then? It is a DNA molecule that is independent of its existence and it can self-replicate and express. So we need our foreign gene or the DNA segment inserted into this vector where it can replicate as well as express. Alright, so it's so interesting here. So the entire lot can be compiled to one tool, that is genetic engineering, which is the manipulation of the plant genome via introduction of a characterized DNA segment. Now, let me put it in a very simple layman's words. You have two plants, plant A as well as plant B, and they have genes X as well as Y. And plant B is the plant of interest for you. It's a crop plant which gives a good yield and so on and so forth, but it is not resistant to high temperature condition. But plant A has an X genes which is extremely resistant to high temperature conditions, and plant A is not of interest for us. It doesn't yield, it's a weed. Let's consider it as. It's a very arbitrary example that I am giving. Now you have transferred. So you are interested in gene X. So you have transferred this gene to plant B. Now B has X as well as Y genes, where X is the high temperature resistant gene, or let's call it as a drought resistant gene. So this transfer of the gene from plant A to plant B, where plant B expresses this gene, that is the X gene, okay, is a transgenic plant. Got it? Well, let us see how useful is genetic engineering in improving agriculture. First, any elite variety can be used to produce a single trait. Second, the genes or the gene that is coding for disease resistance, insect resistance, herbicide tolerance, etc. can be transferred from another organism into the plants. Then we can also think of improving the quality traits like the protein and the carbohydrate content can be modified. The plant oil and the fatty acid composition can be modified. You can enhance the flavor and texture. And also you can increase the longer shelf life of a particular fruit or a vegetable. Now that's very important. It's because for export, value decreases when there is a shorter shelf life of the fruits and vegetables. So this is an advantage when you increase the longer, the longer shelf life of the fruit or vegetable is increased via this technology. Now, what are the main potential benefits of genetic engineering in production of these transgenic plants? Here, it actually increases the nutritional value of the crop, and also there is higher yields from the crop. Okay, and there is reduction in pesticides as well as fertilizer use. See how good it is, see, there is no environmental pollution because continuous use of these chemical fertilizers as well as pesticides, the soil will become very hard. It's not fertile soil anymore, right? So there is always a reduction in the pesticide and fertilizer use, which is one of the benefits of genetic engineering in production of a transgenic plant. And then I did tell you enhanced nutritional value, and a disease resistant plant can be produced, drought resistant plants can be produced, and there are many heterologous molecules can be produced in a plant, in a transgenic plant, which do produce variety of products. Okay, so these are the potential benefits of the genetic engineering in the production of a transgenic plant. Now let us see the strategies for transgenic production. Strategies of transgenic production. Basic concept of gene and DNA traits, identification and isolation of gene for specific traits, designing of gene constructs for transformation, gene transformation techniques, and selection and regeneration of putative transgenics. Basic concepts of DNA and traits. Now here, because of the universal presence of DNA in the cells of all the living organisms, it is possible to construct a transgenic plant. Now here the DNA stores all the genetic information of the organism and it controls the metabolic process of life. Now here the genes, they are the segments in the DNA that encodes a specific proteins. And when these proteins are produced, they function as enzymes and catalyze many biochemical processes, or they are the structural or the storage unit of the cells to contribute to the expression of a specific trait. See, this is the basic thing. This is the general pathway of flow of genetic information. You can see here that the information encoded in DNA is expressed in the form of proteins via an mRNA, an intermediate product. The transcription and translation process, which is indicated as 1 and 2 respectively, are processes which is controlled by a complex set of regulatory mechanisms so that a particular protein is produced only when it is needed. So we can see here that even species that are different from each other, they have a similar mechanism for converting the information in DNA into proteins. So thus a DNA segment from bacteria can be manipulated and translated into a functional protein when inserted into a plant. So this is fantastic, and this flow of genetic information is what allows us to produce a transgenic plant. Identification and isolation of genes for a specific trait. Now here, this is the most limiting step. Here what happens is there is very less information that is available for all the traits that we are looking into, just like enhanced yield of the plant, then stress tolerance, disease resistance, modified chemical characteristics of the harvested products. Okay, now here, once you have identified the trait or the gene for that particular trait,
you also have to see that whether it is going to affect the plant. Secondly, you have to see how that particular gene is regulated, or whether this gene is going to interfere with the active genes of the plant in that particular biochemical pathway. So overall, it should not, uh, this gene of interest should not affect the plant of our interest, right? So if it is going to affect, then there is no, it's going to be a disadvantage. So it has to be completely characterized. There are many new technologies that is available in order to sequence and study the functions of these genes of that particular trait, and then going in for the transgenic production. In the strategies of transgenic production, we finished with the first 2 points. And the next video of mine will show you designing of gene construct for transformation. It will be continued.